only nature, what is happening in nature. I am not going to tell about any of my research or any other research work also. Okay. What is happening in nature, that is the only I am conveying with some slides. Some slides already I mentioned that some slides are all objectionable. Kindly bear with me. That's it. Okay. This is the one when you start about sex. Virtually all metazoans, like multicellular organisms, have sex. It is why sex, but why two types of categories? What makes only two categories of sex? How sex determination in humans is different from other organisms? All these things is meant, it will come to your mind. But nowadays, not only boy or girl, we are also thinking about intersex or transgender. This type of questions, this type of words is coming to you in your mind. In that way, this is the one. What is sex? What determines gender? Gender is different from sex. What determines gender? This type of things we are going to understand exactly, since we are all studying biology and we are teaching biology or science. In that way, this is nothing but is a masala. When you start about sex, it's a masala, different type of ingredients. These ingredients will become the masala. Okay. In that, you can see this is the one. What is sex? In that, you can see this is the standard definition of sex. The sex is the predominant mode of reproduction in multicellular organisms and is primarily responsible for radiation of multicellular taxa. We are having species diversity, genetic diversity, ecosystem diversity. All these things is originated from this sex. Okay. Then another one definition, as per the British Medical Journal, you can see this, despite the fact that the women significantly different from men, there is considerable reproductive evidence that they belong to the same species. We are all belongs to the Homo sapiens, but male is different, female is different. If you take any organism, any species, male is different, female is different. That is the one we call as sex. Since we are all biologists, the biological basis of sex is the sum of those differences in structure and function. If you take male, gonads are different, gametes are different, the genital organs are different, which is organism classified as a male and female. In that, sexes are defined by mutual incompatibility between the same mating type. It's the same mating type. The human's mating type is same, but genitalia is different. Incompatible. It's not compatibility. Structure is different, function is different. That is nothing but biological basis of sex. In that, this is the one. If you take that, most organisms have two sexes. Monoecious species. Hermaphrodites, we also called as hermaphrodites. Some animals like snails, earthworms, and most plants, both type of sex organs are present in same organism. That's why we call as monoecious species. Another one form we call as dioecious species. Some of my plant, biology, botany friends are there in this platform. They have seen that cycas. And in case of date plants, in case of Saudi Arabian countries, they are growing this one. You can see that male plant is different, female plant is different. In case of our cock, peacock, hen is different and cock is different. Okay, this is the one we call as dioecious species. The sex is separated. Okay. In that, this is the one that form sexual dimorphism. Sexual dimorphism we have seen in frequently we have seen in birds. particularly birds, male is different, females are different. Female bird, male birds. You can see this is the one, is a common blue butterfly. Same in case of butterfly. Another, this is our Sayadri birdwing. This is our Karnataka state butterfly. It's named as state butterfly of Karnataka. It's a southern birdwing. You can see another one pair. You can see this is the Bombyx mori in case of silkworm. The male is different and female is different. You can see even in case of Homo sapiens. If you take even this mallard duck, it's a famous. You can see male and females entirely different. Male is different and female is different. You can see this is the one Panthera leo, and you can see this is the one Panthera tigris. This is a very beautiful one. You can see that male and female black buck gesture. The male entirely different from female. Okay. That means the sex is the one is going to form the dimorphism and develop in a formation of diversity in our nature. You can see this is the one is a model we call as traditional binary gender model. So biological sex means hormones are going to secreted, genitalia is there, secondary sexual characters are there. That means male and female we can differentiate based on the biology. If you take gender, gender is nothing but expression. What type of dress we are going to wear, posture and role identity. Some of them are masculine and we can say that feminine. The orientation, if you take that males always attracted towards women, that female is always attracted to the men. This is the one we call as traditional binary gender model. It is there in our nature, in our society, but it will be changed after my talk. At the end of my talk, this model is going to be changed. Okay. In that, you can see this is the one. The next question is, why do we need sex? Why do we need sex? In that, you can see this is the one our Charles Darwin and August Weismann, they clearly mentioned that the purpose of sex was to generate genetic variation. That's hybrid vigor. We need to develop hybrid vigor. For that, we need sex. Okay. In that, this is the one why sex is this is the one. Everything, the changes, the mutation, hybridation, everything is occur at an individual level of change and reproduction rather than the population. For that, we need sex. This is the one at the time of segregation, at the time of meiosis, that can disrupt advantageous combination of genes more often than it creates new, better adapted ones. That's why we need sex. It is going to adapt, it is going to be evolved, then it is going to form a new taxonomy. It's a multicellular taxa is going to form. For that reason, we need sex. In that, the history, just I am going to give you some brief idea about the discovery of sex determination, history of sex determination. The first person you know that is father of zoology or biology, all these things we have to say. Is the person is called as Aristotle. The sex is determined by the heat of the male partner during the intercourse. He was so perfect person. Even though 335 BC ago, he was mentioned that one, still this statement is pertinent to our nature. He is a right person. He is very right person, that Aristotle. Okay. That I will give you the evidences after this, after 5 or 6 slides, how the Aristotle is perfect in this type of sex determination concept. The second one is, this is the one, up to until 20th century, we don't have any idea about sex determination. You can see that environment, people are thinking about that environment, temperature and nutrients. About all these things are going to be influenced the determination of the sex determination. Particularly if the factors like favoring the storage of energy and nutrient the mother is going to store energy and nutrients. She is going to give birth to the females. Whereas other factors favoring utilization of energy and nutrients, the influence want to have male offspring. This type of concept still it is there in our society. People are thinking about, nutrients, temperature, environment, all these things. Still, we are thinking, even though it's a sum of things is scientific, still we are thinking about this type of concept. Only in this, 1891, this, German biologist Hermann, he was pointed out that something is there in the cell that is nothing but is X element, we call it as X body. It could be the reason for determining the sex. Then again, that another one person, American zoologist, McClellan in 1902, he was mentioned that X element, that is nothing but accessory chromosome, is responsible for determination of sex. In that later, these two scientists, one is Stevens and Wilson. Edmund Becker, is Wilson's student, is the Nettie Stevens. She was identified and named as a two chromosome, X and Y chromosome, in these beetles. These beetles are always present in your rice. She observed that chromosome. She has mentioned that the male beetles are going to have X and Y chromosome, that is nothing but heterogametic sex. If it's a female are having XX chromosome, that is nothing but homogametic sex. Nettie Stevens, she was proposed that this hypothesis of sex determination in 1909. Up to 1909, we are not having any scientific evidence of sex determination. The male is formed when egg is fertilized by sperm carrying Y. Female is formed when egg fuses with X carrying sperms. This was the hypothesis, the sex determinant hypothesis proposed by Nettie Stevens. Now we are having this concept of chromosome. Chromosomes are nothing but is a long continuous strand of DNA. It's a plus several types of associated proteins and RNA. It's a metacentric, telocentric, acrocentric, submetacentric, all this thing is there. In that humans, we are having 23 pairs of chromosomes. 22 chromosomes are, up to first 22 chromosomes are autosomes. The last 23 pairs, that is nothing but XX, it is a female, XY is the male. Okay. When it is during this fertilization, particularly most organisms have two types of gametes. Gonads are different from gametes. The gametes are nothing but anisogamy gametes. Anisogamy means large production of large and small gametes. We call it as anisogamy. Particularly females, they are going to produce eggs. That's a bigger one. It's only few, particularly humans, one egg for 1 month, one cycle. That is immobile. They're having lot of resources. But if you take that male gametes, there are numerous, they're small, and they're always motile. That motility is very important at the time of fertilization. Some of the males, they are infertile, means the motility is the major issue. Okay. In that, at the time of fertilization, applied set of sperm is going to fuse with applied ovum, that is egg. You can see that egg and sperm is going to fuse, that is producing a cell with 46 chromosome, 22 pairs of autosomes and one pair of sex chromosome. The one pair of sex chromosome is the one is going to determine sex of the offspring. You can see that sperm that fertilizes the egg as an X chromosome, the embryo will be XX female. The sperm that fertilizes the egg as a Y chromosome, the embryo will be XY. It's a male. In that case, this is the one in our genetic XX and XY system. The male determines sex of the offspring in case of humans. In case of humans, the male determines sex of the offspring. That is nothing but the sperm carrying X fused with egg, X egg, it will become female. The sperm carrying Y, it is fused with egg of the X, it will become the male. XX is a 50% chance, XY is a 50% chance. It's a random event that depends upon at the time of fertilization, it is going to determine. That clearly I'm saying that in case of humans, the males are the one is going to determine the offspring of the sex. In that, now we can come to the X and Y chromosome. We know that now we understood that X and Y chromosome is the one is responsible for determination of the sex. If you take X chromosome, it's a bigger chromosome. It is having many genes, lots of genes. Mostly they're all responsible for many mutations, particularly color blindness, hemophilia, all these things. X-linked one is a markers. They're associated with many mutations. But Y chromosome is very small. It is having very small amount of genes. Y chromosome is very small. X chromosome is long. That is the one I'm going to give you idea in next slide or the end of this lecture. Okay, this is the one. If you see that in the earlier days in the meiosis one, if you take X and X chromosome, they are going to pair. The crossing over is going to be happen. The recombination is going to taking place. But in case of X and Y chromosome, the crossing over is not possible. You can see that no crossover, and X and Y chromosome, the sequence is diverse. Only autosomal reason the crossing over is taking place. If you take this is the one, you can see that in case of XX chromosome, crossing over is taking place, but in case of X and Y chromosome, there is no crossing. You can see this is the one, particularly Y chromosome. They are having this one male specific region on the Y chromosome. You can see that euchromatin region. You can see this is the one no specific to the sex. Okay, no crossing over in case of Y chromosome. No crossing. You can see this is the one. If you take Y chromosome, it contains MSY region. MSY means male specific region. But if you take another one, another part of the Y chromosome, we called as sex determining region of the Y, we called as SRY gene. SRY and MSY gene. Okay, this is the one in that.
Sir, you can ask Sachin Patil to unmute, sir. Sir, mute, sir.
Yeah, thank you. If you see the chromosomal basis of sex determination, already I mentioned that particularly XX is the male, if it is XY is the female. If that fertilization is happened with ovum X and sperm Y, the zygote can become the XY. If it is XY, the Y chromosome is going to have SRY gene. The SRY gene is the one is going to influence the development of testis. The testis is the one is going to develop into the male sexual characters. If you take only X chromosome, O of the X chromosome, sperm of the X chromosome, that is going to fuse to form a zygote XX. But no Y chromosome, no SRY gene, then that will develop into ovaries. The SRY gene is not developed into ovary. Okay, this is the concept we called as chromosomal basis of sex determination. Okay, this is the one, simple idea of sex determination. Just I am repeating again. If ovum of the X chromosome fused with Y chromosome sperm, that will form a zygote XY. The Y chromosome is there means Y is carrying XRY gene. SRY gene is the one is going to influence the development of testis. But in case of zygote is having XX chromosome, no Y chromosome, no SRY, no testis, it is going to develop ovaries. This is the simple idea about sex determination in humans. Still, we will go at molecular level, what will happen at the time of sex determination. You can see this is the one. You are having Y chromosome means that is having SRY gene. That SRY gene is going to trigger the testis formation. You can see SRY gene produces a protein called as testis determining factor, TDF. SRY gene, it is going to produce TDF. TDF is influence the development of gonads to become testis. Once testis is going to develop, it is going to produce testosterone hormone. The testosterone hormone is going to trigger the development of external male organs. This is the one in case of XY zygote. The zygote is having Y chromosome. Y chromosome is having SRY gene. SRY gene is going to produce TDF. The TDF is the influence the development of testis. Once testis is developed, testis is going to produce testosterone. The testosterone hormone is going to influence the development of external male genital organs. In case of XX female, this type of pathway is not there. This type of pathway is not there. This is going to be happen 6 weeks after conception of that. There you go. Okay. Now you got idea. Up to now, you got idea.
Yes, sir.
Yes.
Sir.
Shall I continue
Yes, sir.
further molecular level?
Yes, sir. Yes,
Yes,
sir.
yes.
Yes, sir.
Now we
Very
can
simply
see
you have explained, sir.
Now I mentioned that the zygote is having Y chromosome. Y chromosome is having SRY gene. SRY gene is going to produce the TDF fact, means testosterone factor. The TDF is the one is influence the development of testis. The testis is the one is going to produce the testosterone hormone. Testosterone hormone is the one is responsible for development of male genital organs. Okay, this is the concept. In that time, many scientists started studying to give some evidence. They want to give evidence. That was the hot topic. Hot topic in 1990-2000. This is the one SRY codes for testosterone factor is making the male mice. You can see this is the person we called as Koopman. Koopman is the famous person. He gave molecular evidence of sex determination using mice model. This article is published in Nature. This experiment we called as home run experiment by Koopman et al. Using transgenic mice. Okay, this is a transgenic mice. What he did was he was taken SRY gene. Just I will show you this one. SRY gene. SRY gene is translocated to the X chromosome. X chromosome that will become males. SRY is absent from Y chromosome. You remove the Y chromosome, SRY gene from the Y chromosome, that will become the females. This is the one we call as transgenic experiment. Simple, this is the one. Nuclei fertilized with XXX injected with SRY gene. You know XX egg injected with SRY gene. Then the eggs were transported to surrogate mothers. The SRY gene was then randomly incorporated into a chromosome and was inserted in subsequent cell division. Animals were karyotyped after development into adults. Okay, this is the experiment he did. Simple. This is a XX zygote. SRY gene is injected. It is randomly translated. Okay. Then after that, the embryo is going to be transferred to the surrogate mother. Then it will become actually XX chromosome. That mice will become male. Y means he was injected SRY gene. You understood? Can I repeat again? You can see that the zygote of the nuclei was taken, that is having XX chromosome, just injected SRY gene. It is randomly incorporated with that chromosome. Okay. Then that embryo is transferred to the surrogate mother. That it gives rise to the offspring. The offspring will become male. But karyotypically, the chromosomes are XX. Just I will give you evidence. You can see here is the XY male, XX also male. You can see XX means it's a female. He injected the SRY gene, that will become the male. This is the evidence to show that the role of SRY gene in determining the sex. You can see this is the one, is a gel. In that gel, you can see that XY is SRY gene is there. XX injected SRY. That's why you can see that SRY gene. From this, we confirm that the SRY gene present in the Y chromosome is the one is responsible for determination of sex of the male. Okay. In that, again, another one experiment. This is the one Alfred Just experiment. This also Alfred Just experiment. He is also given some evidence that female fetus, you remove that ovary, that will develop into female adult. Okay. If it's a male fetus, you remove the testis, that will develop into the female. That means that the testis is the one is responsible for development of male sexual organs. The testis is the one is going to secrete testosterone. The testosterone hormone is the one is responsible for development of male sexual characters or male genital organs. This is another one experiment to prove that the Y chromosome is having SRY gene. SRY gene is the one is going to produce the TDF factor. TDF is the one is going to influence the testis. The testis is the one is going to secrete the testosterone. The testosterone is the one is responsible for development of male sexual characters or genital organs. Okay. In that, this is the one everything is going to be happening. This is the pathway. If it is a Y chromosome is there, SRY gene, TDF, testosterone factor is going to produce the gene on the Y chromosome. That is the one is influence the development of the testis. The testis the one is going to produce the testosterone. But in case of female XX, no Y chromosome, no TDF, SRY, no TDF, no testis, no testosterone, no male character, only female characters are going to develop. This is the one is the going to develop. This is the embryonic development of genitalia. How it is going to be happen? Embryonic development. It's a no SRY gene means no testis formation. The female genital organ is going to develop. Only at the time 8 weeks, particularly used to go scanning at 8 weeks means 2 months. After 2 months, after 6 months, 6 weeks of the conception only, this is going to be happen. Usually 8 weeks, 12 weeks, and 16 weeks. 16 weeks at the time of 4th month, you can get idea about exact that particular fetal sex. Then people used to go for feticide and other things. Okay, this is the one under the influence of SRY gene product, gonadal to develop into testis. How this embryonic development is going to take place, that is the one I am going to explain now. You can see here is in embryo, 2 ducts are there. One duct we called as very important. One duct we called as Wolffian duct. Another one duct we called as Mullerian duct. These are the 2 ducts we called as embryonic ducts. One we called as Wolffian duct. Another one we called as Mullerian duct. The Wolffian duct is developed into male genital organs like semenical vesicles, vasa deferens, prostate, epididymis, testis. The male genital organ is going to develop from the Wolffian duct. From the Mullerian duct, female fallopian tube, ovary, uterus, vagina is going to develop from the Mullerian duct. These 2 ducts are very important. With the influence of that SRY gene, these 2 ducts, SRY gene, these 2 ducts is going to be activated. Either one is going to be activated. How it is going to be activated? You can see here. Both are, just I will tell you here, both are undifferentiated. Undifferentiated depends upon the SRY gene. SRY gene is there. That is the one is going to produce testosterone. Then also producing Mullerian inhibiting substance or Mullerian inhibiting hormone. This hormone inhibits the Mullerian duct development. Inhibit the Mullerian duct development, then it will develop only male. androgen, testosterone, Mullerian inhibiting hormone is secreted by your testis. Means SRY gene is there, means testis is going to form, testis is going to produce testosterone and Mullerian inhibiting hormone. Then testis is going to develop, then testosterone secreting Mullerian inhibiting hormone that will inhibit the development of Mullerian duct in case of males. If no testosterone, no Mullerian inhibiting hormone, then female genital organ is going to develop. This is the one you can see here is indifferentiate, depends upon the TDF, the testis is going to say testosterone and Mullerian inhibiting factor. But no TDF, ovaries, no testosterone, no Mullerian inhibiting factor. That is the one, it's going to happen, depends upon the Y chromosome SRY gene. SRY gene is going to produce TDF factor. TDF factor is going to develop testis and testosterone hormone. This is the exact molecular mechanism taking place in humans, in particularly mammals. This is the one you can see all this gonadal differentiation is taking place 6 weeks. Then depends upon the particular gene, their ovaries are going to develop or testis is going to develop. At the time of 20 weeks, means 4 months, exact gonads are going to form. Okay. This is the one you can see the testis, Y chromosome, TDF, testis, leading cell is going to produce testis, testosterone hormone, Sertoli cells are going to produce anti-Mullerian duct hormone. Then male sexual characters are going to develop. In case of ovary, there is no Y chromosome, no TDF, only ovaries are going to develop. Theca cells are there, granular cells, they are going to produce estrogen. Estrogen is going to develop into mammary glands and breast tissue. Then other uterus, oviducts, and other genital organs are going to form in case of females. This is the basic idea about sex determination in humans. This is the continuous process. It's a genetic, gametic, chromosomal sex, gonadal sex, then phenotypic and internal external. Finally, it's a behavioral and metabolic sex. Okay. This is going to be happen. Finally, it is the one leads to intersex. How intersex is going to form? That's a question. Intersex, that is nothing but they used to have both type of sex, both type of genital organs. That is nothing but hermaphrodites. Hermaphrodite, which means that they are having an abnormal number of chromosomal makeup, mixed or intermediate male and female sex characters. Biologically, they are going to have both type of genital organs. Depends upon the their influence, they act as a manly or womanly. Okay. In that case, this is the one we called as androgen insensitivity syndrome. Androgen insensitivity syndrome. It's a mutation in X-linked androgen receptor gene. Androgen receptor gene means testosterone receptor gene. The XY male becomes phenotypic females. They're having XY chromosome, but phenotypically they will become females. You can see testosterone is going to produce, but testosterone receptors are absent. The cells are going to develop as a females. It's a X-linked genetic traits that cause XY individuals to develop into the phenotypic females. This is the one we called as androgen insensitivity syndrome. You can see this is the one is a mild androgen insensitivity syndrome or partial androgen insensitivity syndrome or complete androgen insensitivity syndrome. They're going to have both type of genital organs. You can see here you can see that androgen insensitivity. Because if it is XY, they're going to have testes, but testosterone is also produced. They don't have receptor. That will become phenotypically females. Okay. Then again you can see that this is the one we call as androgen insensitivity syndrome. You can see XY karyotype, internal testis is there, female secondary sexual characters, almost always raised as a females. You can see another one is, this is the one mutation in 5-alpha reductase. 5-alpha reductase. That's the enzyme. Alpha reductase enzyme is responsible for conversion of testosterone into dihydrotestosterone. It's absence of that, they're developed into this. They're going to have intermix. It's a mixed type of genital organs. Okay. Then again, another is congenital adrenal hyperplasia. The testes are absent. They're going to have testes are absent because of defects in steroidogenesis that developed into the autosomal recessive. Then testes are absent. The normal Mullerian structures are developing. Mullerian duct is going to develop into ovary. That is the problem. Okay. This is the one. Another is persistent Mullerian syndrome. It's a male. It's a normal male genital. They are going to have normal male genitalia. The presence of uterus and fallopian tube. Why means some Mullerian duct is going to be retained. That should be inhibited. That should be inhibited. That should not develop, but it is going to be retained. That developed the female is a male is going to have female genital organs. This is the one. It's common. It's common even in case of amphibians. Even in case of amphibians, ovary and testes, you can see it is common in ovary and testes in case of amphibians. Still, I am working on amphibians. We are also seen that this type of mixed type of genital organs is seen in amphibians. Okay. This is the one example. You can see this Kester Semenya. Kester Semenya is a South African middle distance runner. It's hermaphrodites. It's hermaphrodites. She has no ovaries, internal testis, no womb, and ultra high levels of testosterone. She raised as a female, but particularly she caught in the testing. Okay. Again, another one is, this is the one, our Indian one. It's a Shanti Soundararajan. You can see now this is a male. Earlier she was the female. She won many gold medals and everything she won. Then in the test, they found that she is a male. Now he is working as a daily basis. Okay. Another one concept we called as pseudo-hermaphroditism. Pseudo-hermaphroditism. You can see this is the one. You can see that testes are there. Testes are there. Again, that female genital organ. Female genital organ. You can see this is the one. The gonads are testicle. Is a retained. The tubular component is a female. Fallopian tube, but other thing. The external genitalia is a female. This is the one we call as pseudo-hermaphroditism. Okay. You can see this is the one. Is a pseudo-hermaphroditism means they are, XY individuals. They're born as a male, but they develop females. The phenotypically they will become females at the time of puberty. You can see at the time of puberty, females change into males. This is the one we call as pseudo-hermaphroditism. The best example for pseudo-hermaphroditism is this Pinky Pramani. You can see that she won many gold medals, silver medal, everything. You can see that she's a female, it's a girl. You can see here is a male. At the time of puberty, she was also a pseudo-hermaphrodite, one a best example. Okay. Then again another one, this is the pseudo-hermaphroditism. They're going to have both type of sexual organs. Okay. You can see this is the one, another one person. It's a both sex organs, depends upon their wish. They used to go surgery, they're going to remove one organ, they retain one type of organs. This is the one. Recently, this is the one transgender. Last two days or 3 days back, only she has appointed as a first guest lecturer in Karnataka. Okay. This is the now, this is a model we called as modern continuum gender model. It's not like traditional male and female or gender masculine or feminine or sexual orientation is women or men. This is the one is called as intersexed, is androgynous, or some of them are gay, some of them are homosexual, some of them are become bisexual, depends upon their sex, intersex. Okay, this is the one, it is there in our society, in our nature. If you see, this is the one, the person is like this. I scored 96% in 10th, 92% 12th, captain in school cricket team at great time in IIT. Now if I am a gay. So why has it made things so different? Means our our setup, our natural setup. Okay, this is the one. Wherever you go, our network follows like there is no Vodafone or something advertisement. Wherever you go, hatred follow. They are the one group they're going to have in their society now. Okay. This is the one. If you take sex, it's not necessary for all life. Y chromosome is not having any role in some of the organisms. And some of the organisms, they abandon their sex. They are going to produce their offsprings asexually. Okay. In that example, this is the one, the sex of the offspring is written by variety of ways in other species. But in case of humans, it's a genetic or chromosomal base. But in case of other organisms like this one, so birds, is a birds. You can see this is the one ZZ and ZY type. In case of humans, XX and XY. But in case of birds, ZZ means it's a male, ZW means it's a female. Means, but some mother determines the sex in case of birds, mother. But in case of humans, the father is determines the sex of the offspring. Okay. But in case of some butterflies and moths, same ZZ and ZW type of sex determination. But in case of beetles, what Nick Stevens mentioned, XX and XY type of sex determination. But in case of butterflies and moths, the concept is reverse. But in case of beetles, XX and XY. But in case of butterfly, Leptoptera, order Leptoptera is ZZ and ZW type of sex determination. You can see this is the one, another one, honeybee. Honeybee, you can see this honeybee is a sex determination is we called as haplodiploidy type of sex determination. Haplodiploidy. Haploid set of honeybee chromosome is a male honeybee and diploid set of chromosome in case of female is a queen. That is the one 32 chromosome, another one we call as 16 chromosome. This is common in case of ants, bees, and wasps. Okay. You can see this is the one we call as haploid diploid is Hymenoptera. Hymenoptera, no sex or chromosome, no chromosome sets, only male. One set haploid is there, it is going to fertilize with the ovum that is going to produce the fertile female. It is no fertilization that will become the males. Okay. In case of bees, ants, and wasps. You can see this is the one, another one example. Environmental temperature is going to determine the sex of the offsprings. Example is in case of, you can see that turtles, in most turtles, sex is determined by their temperature, where that egg is going to be incubated. The sex is going to determine based on the temperature. You can see these leatherback turtles hatching, they will come out, depends upon the temperature. You can see this is the in case of alligator, crocodile, particularly reptiles. That is the one Aristotle clearly mentioned that the heat of the body is going to determine the sex of the offspring. In case of that, reptiles is a perfect concept. You can see here is many reptiles, no sex chromosome dimorphism. Sex of the offspring is determined the temperature at which the eggs are going to be incubated. You can see that 29 to 31, all will become female. 30 to 33, all will become males. That means that the temperature is the one is going to determine sex of the offspring in case of reptiles. Okay. Same, this is common in case of some insects, that is it's a scaly insects. This is the one, this insect you can see here is when the temperature is warm, that will become the females. When the temperature is very cold, that will become the male. Okay, this is the one we call Taliopora tubulosa. You can see this is the one is stick to the some plants and is going to produce their pupa. The pupa is developing the adult one. Whenever that this adaptive advantage is favoring the production of the female offspring. Okay, depends upon the temperature, they will become female or male. It's another one example is Phycilocyrus pictus. This is the one we call grasshopper. You can see this is the grasshopper. There is no Y chromosome. In case of grasshopper, no Y chromosome. The grasshopper is females are excessive homogametic one, but males are heterogametic X0. This is the one. Another one is genic sex determinant system. So many genes are involved in case of plants, protozoa, fish. No sex chromosome, no Y chromosome. Only multiple genes are the one is responsible for development of particular sex. This is the one in case of Drosophila. This was proposed by Bridge. It's a XX and XY type of sex determination. That is nothing but genic balance system. Okay, this is the one. In that Drosophila, no role of Y chromosome. Only number of X chromosome and the autosomal ratio is going to determine sex of the offspring. In case of Drosophila, no Y chromosome role. You can see this is the one. In case of Bridge balance theory, the sex in Drosophila is determined by a balance between autosomal alleles of the favor maleness and alleles of the X chromosome favors the females. In case of this one, what will happen means in case of XX chromosome, XXL gene is going to be activated. That is the one is activated TRA genes. The TRA gene is the one is going to influence the development of DSX protein that develop into female determinate differentiation genes. In case of no XY gene means there is no X chromosome. That's why XXL not going to be activated. TRA is not going to be activated. That male specific DS protein is going to develop into male differentiation genes. In this Drosophila, there is no role of Y chromosome, but in case of Drosophila, only XX chromosome and the autosome, number of autosome is the one is responsible for development of sex. You can see this is the one, another one beautiful example. This is the one we call as clone fish. It's an ornamental fish. The sex determination is entirely based on the behavior. That is nothing but is called as behavioral sex determination. It's a behavioral sex determination. You can see that in a colony, particularly 2 to 6 clone fish are there in the colony, particularly in sea anemone. The largest fish in the colony is the female. The next largest one is the male. The remaining fish are non-breeding males. In a colony, largest fish is the female. The next largest one is the male. The remaining are non-breeding males. If the female dies, the male becomes the female. That is nothing but it's a behavioral. Once in a colony, female is going to die. The breeding male becomes a female. The next largest becomes the breeding male. This is the one we call as is a strict pecking order. Pecking order. This is based on the behavior. How this is going to be happen means is a concept. This is sequential hermaphrodites. There are hermaphrodites depends upon the behavior, like brain is going to be influenced. The behavior, particular gene is going to be expressed. Depends upon the behavior. Once female is died, the male will become female, that producing the aromatase, then estrogen hormone. This is nothing but is a best example for sequential hermaphrodites. It's called as behavioral type of sex determination in case of clownfish. Then another one example, social influence in the environment. Sex is going to determine by social influence. Another example is a green spoon worm. It's a marine worm. it is a larva is undifferentiated, it's not a male and female. Whenever larva is going to encounter with a chemical signal emitted by the female, it will differentiate it into female. It's a male. If the larva is encounter a chemical signal emitted by female, it will become the male. It's nothing but it's a female is going to determine the male and female of this worm. It's a spoon worm. This is nothing but social influence in the environment. Here also there is no role of Y chromosome or X chromosome. Okay, another one example in case of platypus is a egg-laying mammal. The sex is going to determine by 10 sex chromosome, not one chromosome, 10 sex chromosome, and is also evidence that all these sex chromosome is having our male and some of the higher vertebrate genes. They are sharing with higher vertebrate genes. You can see this is the one karyotype of that platypus. You can see this is the one is echidna. You can see that chicken, platypus, and humans, they are going to share that some of the unique type of genes. That means that we are also, they are the one is a primitive mammal. They are evolved from, humans are evolved from that platypus and echidna. Another one type of example, sex determination we call as cytoplasmic sex determination. In this process, intracellular microorganisms are endosymbiotes that control the sex of their host organisms. This phenomenon is commonly, called as, they are going to change, particularly is a bacteria, Wolbachia is a bacteria, it will change the host sex. It's a feminine genetic males become female phenotypic females, male become females. The host sex is going to be changed by microorganisms. In that, this is the example. You can see that Wolbachia, it is present inside the cells of these insects, invertebrates. They will change the insect sex. Female become males. This is the one we call as cytoplasmic sex determination. Then another one, last type of one sex determination we call as paternal genome elimination. This is present only in scale insects and millibugs. These are all the one, sex determinants. This is the one, another one example, how the mating is taking place in case of anglerfish. This anglerfish, particularly males is going to be merged with, fuse with females. Then they will become single individual. This single individual is the one is the best example for as a natural transplantation. Thus, mating is taking place not by sperm and egg, but they are going to each individual, they will become one. Male and female become one. The male is going to fuse with female, that will become one organism. That is one example for natural transplantation. And also it's histocompatibility. Nowadays, we are calling major histocompatibility, immune response, transplantation rejection, all these things. Is a lesson for our thoughts. Okay, this is the one. How sex chromosome is evolved. Evolved means the sex chromosome earlier, all these sex chromosomes are autosomes, X and Y chromosomes are autosomes. Because of that recombination and deletion of many genes, now we are having X and Y chromosome. In that X and Y chromosome, you can see that evolution of the Y chromosome. Earlier is autosome pair, ancestral reptiles. Now SRY gene is there in 350 million years ago. Now we are having X and Y chromosome in humans. Okay. Now many reports are there. Now we are in future in Y chromosome is also going to be deleted or extinct. In that, these are the models of Y chromosome degeneration. In that model, some of the genes are going to be adapted or accepted, or some of the genes are going to be deleted or expressed. Okay, you can see that earlier autosome 1 and XY were the ordinary pair. The proto X and Y one partner acquires male determining genes, then other male advantage genes are accumulated, the recombination suppressed, then Y is going to degraded, and in future there is no Y chromosome in humans. This is the report frequently we are reading in many new scientific journals articles. Okay. In that, this is the one AA chromosome. You can see that Y and X chromosome. That Y chromosome we are going to last. There we are having only earlier 1,500 genes in case of X and Y chromosome. Now we are having only 130 genes because of the deletion or the removal of that particular genes. Now several rodent species have completely lost the Y chromosome. In the human Y chromosome also we are adding to the extinction, that is nothing but the last man in that. In that, this is the one already even chimpanzee is also going to last that Y chromosome. In that case, what will happen means future of the sex means this is the one, the Y chromosome is particularly vulnerable because it is not matching pattern of the X chromosome. It cannot retrieve genetic information by recombination. So already I mentioned that XX is a recombination crossing over is taking place, but in case of X and Y, no crossing over. That is the only reason they are going to last that Y chromosome. The original contain 1,500 genes. Now we are having only 50 genes. Only 150 to 50 genes is inactivated. That means that at present rate of decay or deletion, Y chromosome will self-destruct in 10 million years. Within another 10 million years, we are going to lose our Y chromosome. In that, the next humans is extinction of humans or Y chromosome. Okay, in that, this is the one, it's like a masala. From this masala, you can make pani puri or sev bhaji or sev puri, anything. This is sex determination in case of humans and other organisms in nature. Okay,